for this session, Santhi Ganesh. Somebody sent me their 10-page hard sketch, and I haven't had a chance to read over it. Sorry. But Santhi's one of those who've become friends over the years, working, collaborating together, and together, we and much of you still here, but we're collaborators on the VanSCAD genetic sub-study, which we received not only funding from the Harvard Foundation, but also from the NIH for Santhi. And Santhi is one of those superwomen. She is incredibly intelligent. Not only she's a cardiologist, but she's a geneticist. She knows all these terms that I have no clue what they are. But she should educate us about that. She has several grants from NIH and JOBIA, you name it. But Santhi, welcome.
Thank you for the invitation to speak. I'm really delighted to be here. We had a really outstanding session this morning, including some setup to some of what I was going to talk about with regards to some of the predisposing factors or associated diseases that have a known genetic basis. So I think what I'll do in my talk today is try to give a very brief introduction to what different kinds of genetic findings can come about from research studies, what's known about from the known arteriopathies, just a very brief primer on genetics, and try to put into context what we have thus far. I've had patients come and ask whether they should be tested, perhaps for the factor 1 gene and things that have come about from the literature, so I was hoping to put that into context for you. My disclosures are really just about grant support related to SCAD. So the outline is general genetics background. We'll go over two different categories of genetic findings, one being monogenic, things that are inherited in patterns that we can describe as autosomal dominant or autosomal recessive, some of the words we heard this morning, or complex genetic diseases, and then some newer genetic findings for SCAD. So very briefly, in terms of the genetic architecture of human disease, when we think about what's called Mendelian diseases, these are the ones that you inherit as one copy from a parent to a child, that would be autosomal dominant, or if you need two copies, that's the recessive scenario. These have a high effect size for alleles or genetic variants that are very, very rare in the population. So this impacts how we approach some of the research and also helps us to think about what kinds of genetic testing are we thinking about, and what makes up the genetic basis of the disease we're looking at. On the other hand, there are very common variants that are higher frequency in the population that have very, very subtle effect sizes. And the idea here is that it's probably not any one of those genetic variants over on that right side circle that is contributing to the disease, but more that there are multiple put together that in a combinatorial kind of way can add to the risk of disease. So examples for these two different categories in the green boxes: familial hypercholesterolemia, Marfan syndrome, which we've heard a little bit about, as a Mendelian type disease, and then coronary artery disease, atherosclerotic coronary disease, diabetes, hypertension. There are some examples that are kind of intermediate, and written in the bold is also the type of genetic study approach that we might take. And all of these have been done in some fashion and are at various stages, and I'll try to put into this framework then what we have thus far. So the current hypothesis of SCAD is that, as we've heard, it's a systemic arteriopathy. There's a genetic predisposition plus inciting factors, and probably there are both rare and common genetic variants, so it's a mixed situation, where in some individuals it might be more that there's a single gene or a single variant driving disease, and in other individuals it might be more of that combinatorial of multiple more common variants. And I'll go through a couple of examples of this. The rare variants are more likely underlying familial forms of SCAD, and there may be possible subgroups of SCAD corresponding to the involvement of different genes. So just very briefly to ask the question, are currently available gene tests useful? And I'll be talking tomorrow a little bit more about the clinical side of things, how to do a clinical assessment. But we've heard this morning about aortic diseases such as Marfan syndrome, which is due to mutations in the fibrillin-1 FBN1 gene, Loeys-Dietz syndrome, which is due to mutations in the TGF-beta receptor pathway genes, and a few other like vascular Ehlers-Danlos to answer this question of whether it could be genetic, first, where we start is with a clinical history and an exam to look for features of an underlying genetic syndrome. And then we look to see, does it essentially fit into a pattern for known genes? If it does, we then might go forward to test for known genes. If not, this is where the gene discovery research comes in, and there's been findings that have come out in the last year. There's more that will be coming. So this was alluded to a little bit earlier, that the question of is SCAD manifestation of known aortopathies or connective tissue disorders. There are 2 studies that are published that have addressed this. One from a Mayo Clinic registry and Massachusetts General Hospital registry, which some of this was presented before, but the bottom line is that a small percent of patients with SCAD do have mutations in these known genes for vascular Ehlers-Danlos syndrome, Marfan syndrome, Lodi-Dietz, and a couple of others. We've done a study looking at whether, kind of the converse, do individuals with thoracic aortic aneurysm and dissection have more frequent SCAD? As you might suppose, if the same genes are involved. We looked at 3 different registries, in total 6,838 patients, and it was really very low frequency of SCAD, surprisingly low actually, I thought, when we had these results. 82% of those with SCAD were female. Major proximal coronary arteries were involved 45.5% of the time. And when, this is one of the more interesting features, that when we looked at which genes were involved, there seems to be a theme here that collagen 3A1 for vascular EDS, TGF-beta receptor 1 and 2, and fibroblin 1 were the ones that were seen. But again, a very small proportion. In thinking about the different subtypes of SCAD and how this might relate back to the underlying genetics, we talked about the monogenic conditions, the familial pattern. This is suggesting a single gene with a large effect. There can be what's called sporadic mutations, which are mutations arising within an individual. So it can still be an allele or variant with high effect, but it's just arising in that individual. But it also opens up the possibility that there are multiple genes with small effects per gene, or possible gene-gene interactions, or gene-environment interactions. Genes linked to multiple arterial diseases, when we see the same thing causing multiple things, that's called pleiotropy. And the best example of this is for the factor 1 genetic variant, where there's a common SNP within the gene. It's not in a coding part of the gene, it's in a part of the gene that's between the coding segments. It's very frequent in the population. About 40% of the population will carry one of the alleles that's associated with increased risk. An odds ratio of 1.4 has been shown for fibromuscular dysplasia, and more recently 1.7 for SCAD. The same exact SNP is also associated with migraine and cervical artery dissection. I know Dr. Hayes showed this image before, and it's from the paper publishing the Factor 1 Association for SCAD. And what's very kind of intriguing about this particular variant is the risk allele for FMD and for SCAD is the opposite allele that is the risk allele for coronary artery disease. And there is a sex difference in terms of the expression, with SCAD being more often observed in women, and myocardial infarction due to coronary atherosclerosis more often in men. So what the biology of that is has yet to be worked out, but very intriguing that we might have the same exact location in the genome increasing risk in different situations to different diseases. Raises the possibility that some of these factors may be protective against other things. So the risk allele that increases risk for SCAD may be protecting from atherosclerosis would be one way to interpret this data. But again, because 40% of the population carries this allele, it's not something that we would test for at this time. An odds ratio of 1.4, 1.7, those are very subtle risks, and a test is not likely to tell an individual about their risk very, very accurately. Just in general, when we go about estimating how much of a genetic contribution there is to a disease, there are a couple of kind of classic ways to do this. One is to look at twin studies, where we look at between monozygotic, so identical twins, versus dizygotic fraternal twins. How often is it that we see that the identical twins have the same exact disease? This is considered a gold standard to figure out what proportion of a disease is due to genetics. The other way to look at this is in a family, look at the siblings and figure out what a relative risk calculation is. And these are currently not available for SCAD. There was a report from Dr. Hayes and her group about familial this may not have been known about in prior generations. I think this is the best evidence that we have, and we're seeing that there are the common variant associations like the factor 1. I wanted to mention a couple of more recent findings because I have had patients come and ask if they should be tested for these genes. So there was a study published from the Mayo group looking initially at a family. So this is often a good way to get a handle on rare variants, family-based studies. And in this family, with the proband, the initial person is the box, the 40-year-old man that had SCAD at age 40. And then his sister had SCAD events at 42 and 44 years. And there was an aunt on the maternal side at age 49 who had a SCAD event. And there's a lot of information you can get from a family pedigree like this. This kind of clustering definitely catches our eye for many of the vascular conditions that we talked about, vascular EDS, Marfan syndrome, Loeys-Dietz syndrome. These are what's called autosomal dominant. You need one copy of the genetic abnormality to potentially have the disease. The wrinkle in that is that there's something called incomplete penetrance. So I'm throwing a lot of words at you, but I want you to know that you can have a genetic variant and not necessarily have the disease. And that's what incomplete penetrance means. You have this disease, but it's not penetrating, it's not showing through the pedigree. And that's where the mother to two individuals has that variant, but does not have a SCAD that is known about. So intriguing, and what further workup of this gene showed through additional sequencing, specifically in the gene, were 10 unrelated individuals with SCAD, and 5 of those individuals had a diagnosis of FMD, which is intriguing and I think raises two possibilities. One is that this is a gene that's a predisposing factor to both FMD and to SCAD, or that FMD or an arteriopathy in general might be at play, and that these are what are called modifier genes. So if you have FMD and maybe you have a genetic mutation in this gene, you're more likely to have SCAD. So there's different ways to interpret that, and it'll be really interesting to see what further information comes out of further study. This is from the same paper, looking at the individuals that were not part of the family. So when we have a genetic variant, we look at seeing the parents often and siblings who has the genetic variant. Wherever there's a plus, that is an individual who carries the variant. And I think what I took away from this is that there were people in the older generations who had the genetic variant but did not have a diagnosis of SCAD. But as we've talked about, there are probably multiple reasons for that possibly being not completely accurate, or this could be, again, that picture of that incomplete penetrance, meaning having a variant doesn't necessarily mean you're going to have SCAD. So the other gene that's been published is TSR1, and I should mention the gene before, TLN1, is something that is expressed in vascular smooth muscle cells. That was part of the derivation and focused on that gene, so a very good candidate gene. TSR1 also was expressed in the arterial wall. This is a study in China looking at 85 individuals with SCAD and nearly 300 controls, and some follow-up, a nice follow-up study of 53 individuals with SCAD and 201 controls. What was unique about this cohort, it was a bit different, with 82% of the cohort being male. And the bottom line of their statistical kind of methods and filtering is that they used statistics comparing the affected cases to controls, and the association level was suggestive. It was suggestive. And so I think what this was published as is a possible locus for SCAD, but more work to be done to really understand this. And further, more work to be done to understand, is this something that is perhaps ethnicity specific or different because of its higher occurrence in male SCAD patients. So this would be a good summary of our current understanding of the genetics of SCAD, that there are some genes known for connective tissue disorders. There are some newer familial findings, but a lot that remains to be sorted out. And again, I'm going to talk about this more tomorrow, so I think I'll save it. But when we talk about genetic screening, just wanted to point out that the gene tests that we usually are able to order clinically are these targeted sequencing panels. So if you look on this list, column 3 of 1 is on both of them, fibrillin 1 is on both, TGF-beta receptors are on both of these panels. These are the most common ones. There are other companies that do it. But the majority of the genes on these panels are aortopathy genes. a point I'll emphasize again tomorrow is that when we're working out somebody with an aortic condition, so a vascular disease that we think is genetically mediated, what we call our pickup rate, our chance of finding something on this kind of testing is probably still in the 20s in terms of the percentage. So only 20 or so percent will be identified on this kind of testing. That's what makes family history and other clinical evaluation very important. So again, just to summarize, we're looking at things that span the spectrum of all the way from rare to common. The rare things being possibly more predictive, but with some caveats, as I tried to outline with the penetrance issue and possibly some of these rare things being modifiers to an underlying arteriopathy, all the way to the common disease associations that tell us a bit about the genetic architecture of these diseases. So in conclusion, there's still a lot more to learn about the genetics of SCAD. The initial findings support both a complex genetic basis and monogenic etiologies. A minority of cases, probably less than 10%, appear to be due to pathogenic variants in genes that are already known about to be associated with connective tissue disorders, as Dr. Wood summarized very well this morning. And most of the SCAD genetics that we think might be going on remains to be discovered.